Good morning, everybody. So now we are going to discuss about microbial growth. In previous time, we have discussed the microbial growth and its media compositions, the microscopic count, method for viable count, and turbidimetric measurement of the microbial cell number. And now we will going to discuss the microbial cell growth and its kinetics. So how the microbes is actually grown, what is the mechanism, the will discuss, followed by what is the about the binary fissions is a asexual reproductions by a separation of the body into two new bodies. In the process of binary fissions, an organism duplicates its genetic material or deoxyribonucleic acid and then divides into two parts. It's called cytokinesis. And with each of the new organism receiving one copy of DNA. And this is the all about the binary fissions by which a bacteria can getting doubled. So if you see in the left side image how the binary fission in a rod-shaped bacterium, here we can see the cell first elongated, and after elongations it will the septum formation is done, and after the septum formation is completed, then cell is getting into two. So when one cells eventually separates to form two cells, we say that one generation has occurred, and the time required for this process is called generation time or doubling time. So as you look in the right side of the slide, we will find the details about how this separation actually occurred. The first step, we can say the young cells at an early phase of cycles, and after that, a parent cells prepares for divisions by enlarging its cell wall, cell membrane, and overall volume. Next, the septum begins to grow inward, and the chromosome moves towards opposite end of the cells, and other cytoplasmic components are distributed to the developing cells. Next, the septum is synthesized completely through the cell center, and the cell membrane patches itself so that there are two separate cell chambers, and at this point the daughter cells are divided and some species separate completely as shown here, while other remain attached, forming chains, doublets, or other cellular arrangement. This is the overall process or mechanism by which bacteria can getting doubled. And very important here, what is binary fissions, and very important to learn here what is generation time or doubling time. Next, we are already talking about the septum formations in the previous slide. So how the septums is look like, the septum here we can see the pictures, that septum that separates dividing the cells of the bacterium is, as per example here is a Bacillus subtilis, is clearly visible in this series of fluorescent microgram. Panel A is the pictures in the left side of the pictures. Panel A, it is stained with DAPI stain. DAPI stain. DAPI is the fluorescent dye, and DAPI fluoresces in blue region. Here you can see the DAPI stain of the entire cells. And panel B is the green fluorescent protein lights up the entire cells. And panel C, a dye that contains only the cytoplasmic membrane, so that the septa contains membrane material. And if you see the membrane material, you can clearly visible the septum of the dividing cells here. And if you think about the growth of the cell cycles, so in the right side you can you will learn details in the course by about the molecular mechanism or replications these bacteria. But here is a small brief introduction of the cell cycle of growing E. coli here. One is origin of replications. Then the bacteria is actually continue to grow by the replisome, and after going replisome, the chromosome getting separated and cell will be separated. These are very brief pictures, but so we have learned what is septa, and we are saying in the visible septa in this slide also. And in the below, there is electron micrograph images of Bacillus subtilis. Bacillus subtilis is a gram-positive bacteria, and here is a dividing and magnified images. Here you can see the electron micrograph images here, the membrane, clearly visible, the membrane, the wall, and the DNA. So the bacteria can grow in binary fission by mechanism, and we'll learn how can we count the bacteria. But now we need to understand its growth cycles, and how the growth cycle is, and how the growth cycles is applicative to ourselves. The microbial growth cycles is maintain a typical curve, and this curve is called bacterial growth curve. If you see in the images, you will find a graph, and the graph is containing lag phase, the graph is containing exponential phase, the stationary phase, and the death phase. And the viable measure of the cells in cultures, the viable measures of the cells in the cultures that are capable of reproducing. And as per optical density, we have learned previously that what is optical density and how can be measured. That is called turbidimetric measurement. And optical density is a quantity measurement of the light scattering by liquid cultures and increase with the cell number. When you are taking a cell, some amount of cells, and you allow for grow, the cell number will be double due to generation time, and then cell will be grow, and the turbidity will be increase, and optical density will be increase. And if you grow the cells, and if you plot the data of the log number of viable cells versus time, you will find the graph like this. And we are also here is that the two graph is actually showing with respect to time is a log number of viable cells and optical density of that bacterial populations. So in growth curve, we are finding lag phase, we are growth curve, we are finding log, stationary phase, and death phase. But this growth curve actually influenced by nutrient concentrations, and the nutrient concentrations having effect on its growth. Now we'll learn how these nutrient concentrations affect microbial growth. Here we can see the graph. There are two graphs in the slide. One is nutrient concentration versus total growth, that is cell numbers or cell numbers, and one is nutrient concentration by growth rate per hour or per minute, per like etc etc. So when microbial growth is limited by low concentration of a required nutrient, the final net growth or yield of the cells increase with the initial amount of the limiting nutrient present, and that is depicted in the figure A. And this is the basis of microbiological assay for vitamin and other growth factor. And the rate of growth also increase with the nutrient concentration that is expressed in panel B. Here you can see that upon nutrient concentration increase, the growth rate becoming is, but it will be touching a saturations after nutrient concentration increasing. But so we can say it's the rate of growth also increase with nutrient concentration, but in a hyperbolic manner, and much like that the seen with many enzymes. The shape of the curve seems to be reflect the rate of nutrient uptake by microbial transport proteins. At sufficiently high nutrient levels, the transport system are saturated, and the growth rate does not rise further with increased nutrient concentrations. Here we can see, and this is the significance of this curve, the panel B. And this is the overall nutrient concentration effect on microbial growth. Now we will going to discuss about the loss of viability. So loss of viability means loss of populations. Loss of populations. So what is so here there is the starvation or growth arrest. There are three pictures: panel A, panel B, and panel C. Okay, so bacteria, whenever you are culturing some bacteria will grow, will maintaining first lag phase, after followed by lag phase they will be in log phase, then stationary phase, and then is a decline phase. In the decline phase, you can see there is decline phase means death of number of cells, and that can be expressed by loss of viability. Okay, so but we will discuss how this loss of viability actually occurred. Okay, so there is some hypothetical theories behind that, and we can explain by this the loss of viability by these theories. The number one, look at the number one. The number one, this if the bacteria in a death phase, there will be some survivors will be present. Some survivor will be present. That is depicted in the figure left side. So it has long been assumed that as cells leave stationary phase due to starvations or toxic waste accumulations, and the exponential decline in the culturability is due to cellular death. Is due to cellular death. The culturability will be decreased. And next point is there will be some belief that a fraction of microbial populations dies due to activations of program cell death genes, and the nutrient that are released by dying cells supports the growth of other cells. So here the panel B, here we can see that genetically programmed cell death, yes, is activated suicide system, and we will get some survivors that consume nutrient leaking from dead siblings. So we can explain by this phenomena. And number three is genetically programmed sterility, and you can call it in short is VVNC, that is called viable but not culturable. So what is the hypothesis of VVNC? The viable but not culturable hypothesis points that when cells are starved, they become temporarily non-culturable under laboratory conditions. When exposed to appropriate conditions, some cells will remain the capacity to reproduce. So we can explain or we can discuss that microbes are temporarily unable to grow. So what is the VVNC? The microbes are temporarily unable to grow, at least under the laboratory conditions used. And this phenomena in which the cells are called viable but non-culturable. In a short, it's called VVNC, is thought to be the result of genetic response triggered in starving stationary phase cells. Just as some bacteria form spores as a survival mechanism, and it is argued that the others are able to become dormant without changes in morphology, and that is depicted in the figure C. So how the viability become lost, we can explain by these three theories. Now, what about the decline phase? What about the decline phase? We are already seen that there is a decline phase in the growth cycles. So if you see in the decline phase, if you see the graph here, the time versus cell number, you can see the overall population is expressed by the express here, and the long-term growth experiment reveals that an exponential decline, the long-term growth experiment reveals that an exponential decline in the viability is sometimes replaced by a gradual decline in the number of culturable cells. So this decline can last months to years. And during this time, the bacterial populations continuously evolve so that actively reproducing cells are those best able to use the nutrients released by their dying and best able to tolerate the accumulated toxins. And this can be explained, this dynamic process is marked by successive waves. And this wave, look at the pictures. You can see there is a successive waves. And these waves is the dynamic process is marked by successive wave of genetically distinct variant. And thus the natural selection can be witnessed within a single culture vessels. So this is the instead of distinct death phase, we can see the successive waves of genetically distinct subpopulations of microbes better able to use the release nutrient and accumulation toxin survive. And then each successive solids or dashed curve represents the growth of new subpopulations. Now we'll going to going to discuss the details about the mathematics of the growth, like it's called kinetics. This is very important. So the knowledge of the microbial growth rates during the exponential phase is indispensable to microbiologists. This is very important for not only the research purpose but also in industry or in economic purpose. Growth rate studies contribute to basic physiological and ecological research and are applied in industry. The quantitative aspect of exponential phase growth discussed here apply to microorganisms that divided by binary fissions. During the exponential phase, each microorganism is divided constant intervals, and thus the populations will doubled in number during a specific length of time called generation time or doubling time. Here you can see a table. Here is an example of exponential growth. Look at the time, the division number and its kinetics. If you, the situation can be illustrated with the simple examples. Suppose that a culture tube is inoculated with 1 cells and that will divide it every 20 minutes. So how the number of the cell will be there? In up at 0 minute, there's population will be 1. At 20 minutes, population will be 2. At 40 minutes, population will be 4. At 60 minutes, population will be 8. At 80 minutes, population will be 16. At 100 minutes, population will be 32. That means the population will be 2 cells after 20 minutes, 4 cells after 40 minutes, and so on and so forth. Because the population is doubled every generations. And the increase in populations is always like 2n. So n is the number of generation. So if you see here the 2 to the power 0, like the number of division is 0, that means 2 to the power 0 is 1. If the number of division is 1, it will be 2 to the power 1, it will be 2. If the division number will be 3, then the population will be 2 to the power 3 will be 8. And if it is division number 6, then population will be 2 to the power 6, it will be 64. So the population will be 2 cells after 20 minutes, 4 cells after 40 minutes, and so forth, because the population is doubling every generation, and this increase in population is always 2n, where n is the number of generation, and resulting population increase in exponential or logarithmic phase. So we can express this one in a graphically. We can see the pictures here where the x and y axis and where is minutes of inoculations versus number of cells, that is red line, and blue line log number of cells. So you can see, if you compare with number of cells, only the number of cells with minute of incubation, the line is look like not straight, it's a hyperbolic. Whereas if you take the log number of cells, you can see the straight line. So we will discuss later how it is significant to take log and how can we express in mathematical equations. So to know the equations, so we can explain like this. The observation can be expressed as equations for the generation time. Let n0 the initial population number. Let n0 is the initial population number. Nt is the population at time t. N the number of generation in time t. So, so, so N0 is the initial population, Nt is the population at time t, N is the number of generation in time t. So we can write the inspection of the result from the previous table. So we can write Nt is equal to N0 into 2 to the power n, because Nt is the initial population at time t, and N0 is the initial population, and n is the number of generations. So solving for n, the number of generations where all logarithms are to be the base 10. So if you take log from the in the equation nt is equal to n0 into 2 to the power n, it will be log nt is equal to log n0 plus n log 2. So from here we can find n. So n is equal to log nt minus log n0 by log 2. So we can write log 2 value. We know the log 2 value is 0.301. So n will be a log nt minus log n0 by 0.301. The rate of growth during the exponential phase in a batch culture can be expressed in terms of mean growth rate constant, and that will be expressed in K. So this is the number of generations per unit time, the growth rate. So it can be often expressed as the generation per hour. So K is equal to, we can write K is equal to N by T. So where is we can write K is equal to N by T, that is equal to log NT minus log N0 by 0.301 T. Here the time is taken the population to double in size. That is the mean generation time or mean doubling time G, and can now be calculated. So if the population doubles, like T is equal to G, generation time is actually the growth time. So we can write NT is equal to 2N0 from the previous equations. NT is equal to N0 into 2 to the power N. Okay. So if substitute to N0 into mean growth rate equation and solve for K, the K is equal to, we can write log 2N0 minus log N0 by 0.301 G. So how can we write here? We can put NT is equal to 2N0. So in the previous equations, K is equal to log NT minus log N0 by 0.301 T. Here NT will be replaced by N0, 2N0. So we can get, if you put 2N0 in place of NT, we will find K is equal to log 2 plus log N0 minus log N0 by 0.301. Log N0 log N0 will be cancelled out, and log 2 will be there 0.301. So we can write K is equal to 1 by G. The mean generation time is the reciprocal of mean growth rate constant. So from this derivation, we can find the final populations by the equation nt is equal to n0 into 2 to the power n. We can find specific growth rate, K is equal to N by T, or we can find generation time from specific growth rate is K is equal to 1 by G, right? So G is equal to 1 by K. So that means the mean generation time G can be determined directly from a semi-logarithmic plot of the growth curve. Here we can see the semi-logarithmic plot of the growth curve. And the growth rate constant calculated from the G value. How can we calculate this growth rate constant from G value? Here is the example. The generation time also be calculated directly from the previous equations. For examples, suppose that a bacterial populations increase from 10 to the power 3 cells to 10 to the power 9 cells in 10 hours. So how the generation time will be? What is the generation time will be? So the cell population from 10 to the power 3 cells to 10 to the power 9 cells in 10 hours. So K will be log 10 to the power 9 minus log 10 to the power 3. So it will by 0.301 by 10 hours. So log 10 to the power 9, it will be 9, and log 10 to the power 3, it will be 3. And in the below, 0.301 into 10, it will be 3.01 hour. So it will be K is equal to 2 generation per hour. So K is equal to 2 generation per hour, the specific growth rate. So now we can calculate G, because we know G is equal to 1 by K. So G will be 1 by 2 generation per hour, and it will be 0.5 generation, 0.5 hour per generation, and it will be 30 minute per generation. So here is the solve. The generation time will be 30 minute per generation. So bacterial populations will be increased from 10 to the power 3 cells to 10 to the power 9 cells if in 10 hours the generation time will be 30 minutes per generations. And this generation time can also determine this is the mathematical equation, but this generation time can also determined from a microbial growth curve. The population data are plotted with, if you plot population data, if you plot population data with logarithmic axis used for number of cells, the time to double the population number is then read directly from the plot. And the log of the population number can also be plotted against the time on regular axis. So this is the another representation of the calculation of microbial growth parameters. Is the same as previous, but here we can jot down the formula. Is like we can jot down the formula is like G is equal to T by N. We can say G is equal to T by N, where G is the generation time and T is the total time for growth. N T is equal to N0 into 2 to the power N. N is equal to log NT minus log N0 by 0.301, or we can see, we can use NT is equal to N0 e to the power KT, or K is equal to 0.693 by G. So we'll discuss now the last two formula, where NT is equal to N0 e to the power KT and K is equal to 0.693 by G. How can it come, right? So as an example, consider actual growth rate from the plot from previous slide, the pictures in which, if as example like NT, if NT is equal to 10 to the power 8 and N0 if 5 into 10 to the power 7 and T is equal to 2 hours, then we can write in the formula like N is equal to log of NT minus log of N0 by log 2, and N is equal to, we can solve this, you can find N is equal to 1. Thus, in this example, if N is equal to 1, and so we can use the equation G is equal to T by N to determine G, that G will be 2 hours. If exponential growth continue for another 2 hours, the cell number would be 2 into 10 to the power 8. 2 hours later, the cell number will be 4 into 10 to the power 8. 4 hours later, it will be goes on, right? And the value of the G can also be calculated by evaluating the slope of the line on the semi-log plot of exponential growth. On such plot, the slope can be defined as log NT minus log N0 by T. Combining this expression and understanding that G is equal to T by N, the slope of the curve reduced to log 2 by G, or simply 0.301 by G. So in this example, which is depicted in the figure in the previous slide, the slope is actually 0.15 per hour. And since G is equal to 0.301 by slope, and the generation time will be, from this equation, we can find generation time will be 2 hours. Okay. So this is actually specific growth rate. That's what is growth rate. So we can say what is specific growth rate. So specific growth rate can be expressed in terms of K, and we can find specific growth rate can be expressed is like directly 0.693 by G. Now, as per example, let to solve a problem. Okay. So, because we have already learned the bacterial kinetics, the mathematics of bacterial kinetics, and the equations. Now we need to solve a problem, because if you solve the problem, we will understand greatly the mathematics or kinetics of the bacterial growth. So, as for example, like here, if one starts with 10 to the power 4 cells in a culture that has a generation time is 2 hours. So how many cells will be in the cultures after 4 hours, after 24 hours, and after 40 hours, 8 hours? Use the equations x or nt is equal to 2 to the power n is equal to n0 previously. Here it is written, here is x is equal to 2 to the power n is x0. Is same as previously, like nt is equal to n0 into 2 to the power n, where x0 is the initial number of cells and n is the generation time and x is the number of cells after n generations. So we can write after 4 hours, n will be n is equal to 4 hours by 2 hours per generation. It will be 2. So x is equal to 2 to the power 2 into 10 to the power 4. So that means 4 into 10 to the power 4 cells after 4 hours. After 24 hours, n is equal to 12 generations. So we can write x is equal to 2 to the power 12 into 10 to the power 4. So 4.1 into 10 to the power 7 cells. And after 48 hours, n is equal to 24 generations. So we can write x is equal to 2 to the power 24 into 10 to the power 4 is 1.7 into 10 to the power 11. So the question, look at the question again. So if we start with a 10 to the power 4 cells in a culture that has a generation time of 2 hours, how many cells will be in culture after 4 hours, 24 hours, 48 hours? It will be the number of cultures by using the using the formula nt is equal to 2 to the power n into n0. So we have found that after 4 hours, the cell number will be 4 into 10 to the power 4 cells. After 24 hours, the cell number will be 4.1 into 10 to the power 7 cells. And after 48 hours, the cell number will be 1.7 into 10 to the power 11 cells. And this represents an increase of less than 1 order of magnitude for 4 hours culture, 4 order of magnitude for 24-hour culture, and 7 order of magnitude for 48-hour culture. And the last, we are talking about what is generation time, what is doubling time. Here is a list of the example of different microorganisms and their generation times. As for example, Escherichia coli is a generation 0.35 hours. That means 35 minutes. Bacillus subtilis is little bit more. It's 0.43 hours. Like Streptococcus aureus, it's 0.47 hours. Okay, Mycobacterium, it's almost 12 hours to grow. As for example, like fungi, Saccharomyces cerevisiae, it will take 2 hours. So here like the examples of generation times. Thank you. Thank you for these sessions.